अब शुरू होने जा रहा है बायोटेक्नोलॉजी एंड इट एप्लीकेशन की सबसे बड़ी डिस्कशन। जी हाँ दोस्तों, इससे पहले कि आपका सवालों का सिलसिला शुरू हो जाए, जल्दी से एक बार बायोटेक्नोलॉजी की 12th चैप्टर है बायोटेक्नोलॉजी एंड इट एप्लीकेशंस में 12.1 जो टॉपिक है, जल्दी से रिवाइज कर लीजिए। उसको रिवाइज कर लिए तो चलिए हम लोग शुरू कर देते हैं लाइन बाय लाइन एनसीईआरटी क्वेश्चंस। तो चलिए बताइए, बायोटेक्नोलॉजी एंड इट एप्लीकेशन में कितने टॉपिक्स हैं? जी हाँ, बायोटेक्नोलॉजिकल एप्लीकेशन इन एग्रीकल्चर, बायोटेक्नोलॉजिकल एप्लीकेशन इन मेडिसिन, ट्रांसजेनिक एनिमल्स, इथिकल इश्यू, ये सारे तो टॉपिक है ना है। तो चलिए स्टार्ट करते हैं। फर्स्ट क्वेश्चन फ्रॉम फर्स्ट लाइन ऑफ एनसीईआरटी डैश डैश एज यू would have learned from the previous chapter essentially deals with industrial scale production of dash dash and dash dash using genetically modified microbes, fungi, plants and animals. The next, what are the applications of biotechnology? The next, three critical research areas of biotechnology are the next। ये बताइए, डाउन स्ट्रीमिंग प्रोसेसिंग टेक्नोलॉजीज किसकी प्यूरिफिकेशन करती है? The next, human beings have used biotechnology to improve the quality of life, especially in the field of dash dash. The next, point 12.1 बायोटेक्नोलॉजिकल एप्लीकेशन इन एग्रीकल्चर। इससे पहले कि सवालों का सिलसिला शुरू हो जाए, प्लीज रिवाइज इट। तो चलिए क्वेश्चन शुरू करते हैं। द फर्स्ट क्वेश्चन फ्रॉम द फर्स्ट लाइन ऑफ एनसीईआरटी। नीचे दिए गए फूड प्रोडक्शन को इंक्रीज करने वाली तरीकों को बताएं। जी हाँ, the next question from the next line of NCERT. The Green Revolution succeeded in dash dash the food supply, but at it was not enough to feed the growing human population. The next, increased yield have partially been due to the use of improved dash dash varieties, but mainly due to the use of better management practices and use of agrochemical fertilizers and pesticides. The next, however, for farmers in developing world, agrochemical are often too expensive and further increases the yield with existing variety are not possible using conventional breeding. The next, is there any alternative path that our understanding of genetics can show so that farmer may obtain maximum yield from their field? Is there a way to minimize the use of fertilizers or chemicals so that their harmful effect on the environment are reduced? Use of dash dash modified crop is a possible solution. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called dash dash dash. What is the full form of GMO? The next, GM plants कैसे यूजफुल हैं? The next, in addition to these uses, GM has been used to create tailor-made plants to supply alternative resources to industries in the form of starch, fuel, and pharmaceuticals. The next, some of the application of biotechnology in agriculture that you will study in detail are the production of pest resistance plant, which could decrease the amount of pesticide used. ये बताइए कौन सी टॉक्सिन प्रोड्यूस्ड बाय ए बैक्टीरियम कॉल्ड बैसिलस थोरेंजेनेसिस। बैसिलस थोरेंजेनेसिस को शॉर्ट में क्या कहा जाता है? The next dash dash gene have been cloned from the bacteria and been expressed in plants to provide resistance to dash dash without the need for insecticides. In effect, created a dash dash. The next examples are BT cotton, BT corn, rice, tomato, potato, and soybeans. The next BT cotton, BT cotton टॉपिक को रिवाइज कीजिए। चलिए शुरू करते हैं बीटी कॉटन टॉपिक से। लाइन बाय लाइन एनसीईआरटी क्वेश्चंस। यहाँ सम स्ट्रेंस ऑफ डैश डैश प्रोड्यूस्ड proteins that kill certain insects such as dash dash, comma dash dash and diphterans, that is flies, mosquitoes. The next dash dash forms protein crystal during a particular phase of their growth. The next biotechnology forms protein crystal during a dash dash phase of their growth. The next, these crystals contain a toxic dash dash protein. The next, why does this toxin not kill the bacillus? The next, actually the dash dash protein exists as inactive protoxin, but once an insect ingests the inactive toxin, it is converted into an active form of toxin due to the alkaline pH of gut, which solubilize the crystals. The next, the dash dash toxin binds to the surface of midgut epithelial cells and create pores that cause dash swelling and lysis and eventually cause death of the insect. The next, a specific dash dash genes were isolated from Bacillus thuringiensis and incorporated into the several crop plants such as cotton. अब cotton रे। The next, the choice of genes depends upon the crop and a targeted pest, as most Bt toxin are insect group specific. The next, टॉक्सिन किसके द्वारा कोड किया गया रहता है, उसका नाम क्या है? The next, there are a number of them. For example, the protein encoded the genes dash dash and dash dash control the cotton bollworm. That of cry dash dash controls corn borer. चलिए 12.1 यानी कॉटन बॉल का माइंड कैप्चर कर लीजिए। नेक्स्ट हम लोग शुरू करते हैं पेस्ट रेजिस्टेंट प्लांट। जल्दी से इसको रिवाइज कर लीजिए। तो चलिए शुरू करते हैं पेस्ट रेजिस्टेंस प्लांट्स। सेवरल डैश डैश पैरासाइट्स, ए वाइड वैरायटी ऑफ प्लांट एंड एनिमल्स इंक्लूडिंग ह्यूमन बींग्स। The next, a nematode dash dash infects the root of tobacco plant and causes a great reduction in yield. The next, a noble strategy was adopted to prevent this infestation, which was based on the process of dash dash. The next dash dash takes place in all eukaryotic organisms and as a method of cellular defense. The next, this method involves dash dash of a specific mRNA due to dash dash molecule that binds to and prevent translation of the mRNA, that is slicing. The next, the source of this complementary dash dash could be formed from an infection by viruses having RNA genomes or mobile genetic elements, that is transposons that replicate by an RNA intermediate. The next, using dash dash nematode specific genes were introduced into the host plant. The next, the introduction of dash dash was such that it produced both sense and antisense RNA in the host cells. These two RNAs being complementary to each other, formed a dash dash that initiated RNAi and thus silenced the specific mRNA of the dash dash. The next, the consequence was that the parasite could not survive in a dash dash host, expressing a specific interfering RNA. The next, the transgenic plants therefore got itself protected from dash dash. फिगर 12.2 का माइंड कैप्चर कीजिए, माइंड कैप्चर ऑफ 12.2. थैंक यू। आपका स्वागत बायोटेक्नोलॉजी एंड इट एप्लीकेशंस के नेक्स्ट सेशन में दिल की गहराइयों के साथ किया जाता है।